All right, here's your lecture for the second half of cardiovascular. In this lecture, we're going to talk a little bit about heart physiology. So that is going to deal with how the heart rhythm and rate works. And then we're going to talk also about the blood vessels. So how is our heart rate regulated? So initially, we receive nerve signals from the brain. And these signals can cause really rapid changes. This is controlled by our autonomic nervous system or the medulla oblongata. I hope you're remembering some of this from when we covered neuro. We have hormones like adrenaline and epinephrine that can target the heart and cause more sustained changes. We talked a little bit about that in the endocrine system. So as far as the heart conduction system, remember this is just a specialized group of cardiac muscle cells in the heart that are going to send electrical signals to the other heart muscles, causing them to contract. We have the SA node that's going to be found in the right atrium. This is going to be the start of the system. So this is going to be the node that actually generates the electrical signal. We refer to this as the pacemaker of the heart. We also have the AV node. The AV node is going to be found between the atria and the ventricles. This is going to slow the signal down so that the ventricles contract after the atria, so that this blood doesn't just end up in a pump that isn't really efficient or functional. We refer to the AV node as being the pace setter. So here is just another thing to help with your studying that shows what this looks like on an actual heart monitor or on an actual heart diagram, sorry. So this number one spot right here, this is your SA node. Again, this is what's generating those impulses and being the pacemaker of the heart. The impulses are going to pause for about 1/10 of a second at the AV node. And then the AV bundle is going to connect the atria and the ventricles to kind of synchronize those. Then it is going to move into the bundle branches. So the bundle branches are going to conduct these impulses all through the interventricular septum, which is this middle piece that runs through the middle here to separate your ventricles from each other. At the very end, it's going to move into these Purkinje fibers. Your Purkinje fibers are going to depolarize the contractile cells for both of the ventricles, and then the whole process starts again. Let's talk a little bit about EKGs, and a lot of everything we're going to cover from here on out we covered in lab, so this should be a review. So whenever you're looking at an EKG, we have here what's called a P wave, then this goes down into Q, R, S, and then a little space, and then a T wave. I love this diagram because it's showing you exactly what is happening at each step. And the whole thing is considered a cardiac cycle. So when your P wave is showing up on the EKG, that is your atria contracting. Also we call systole. And our ventricles are going to be relaxed at that point. Remember, we call this ventricular diastole. The QRS complex, so 2, 2, 2, is where your ventricles are contracting. So at that point, your ventricles are in systole and the atria relax. So your atria are in diastole. So the top here is showing you what your atria are doing, and then the bottom is showing you what your ventricles are doing, and then this is showing you that this is one cardiac cycle. Your T wave, that is just showing you that your ventricles are relaxing, and then again, the whole process starts again after this. So the cardiac cycle is going to be that we just talked about. So that full series of steps from the atria contracting all the way down to the ventricles contracting to the ventricles relaxing is one cardiac cycle. Remember that systole is going to be that contraction phase, and diastole is going to be the reaction phase or relaxation phase. Sorry, I'm talking too fast and getting a little tongue-tied on this one. I will try to slow down. So let's talk a little bit about vital signs. Pulse is going to be the expansion and recoil of an artery with each beat of that left ventricle. So as the blood is being pushed through there, it is stretching on those arteries, and you're actually able to feel that, and that is how pulses happen. A normal resting pulse is usually about 70. A safe one is between 60 and 120. Blood pressure is going to be the amount of pressure on your blood vessels as your heart is pumping that blood through there. It is written as systolic over diastolic. The average is about 120/80. And remember, systolic, we're going to have higher pressures when those ventricles contract. And the diastolic is going to be the lower pressure because this is more when the heart is relaxing and refilling with blood. Let's talk a little bit about blood pressure imbalances. So high blood pressure, we probably all know someone with this or have it ourselves. In the medical community, it's called hypertension. This is going to be too much pressure on the heart and blood vessels, and over time it can cause damage to the heart and blood vessels because they're just being stretched beyond their limits. Atherosclerosis is when blood vessels harden because of that constant increased pressure. And an aneurysm is when blood vessels kind of balloon out and can eventually burst because they're just filling up like a balloon because of that. All right, let's talk a little bit about how we measure how well the heart is doing. You will hear a lot of these measurements talked about if you're working in cardiology or in a hospital. So cardiac output is going to be the amount of blood pumped out by each ventricle in 1 minute. Stroke volume is going to be the amount of blood ejected by a ventricle with each heartbeat. So on one of those, we're looking at a unit of time, whereas the other one, we are looking at the magnitude of how much the heart is actually pumping with each contraction of those muscles. There's going to be 3 different factors that are going to influence stroke volume: preload, contractility, and afterload. Preload is going to be the amount of stretch of the cardiac muscle cells. This is strongly related to venous return and how effective that venous return is. Contractility is going to be the ability of cardiac muscles to generate force. This is going to be largely dependent on if we have enough calcium in those muscle cells and if that muscle tissue is healthy or if it is damaged. Afterload is going to be the force of the blood pressing back on the valves of the ventricles. So the ventricles have to kind of overcome this pressure to pump the blood out. All right, now let's spend just a minute talking about our blood vessels. So we're going to have arteries, we're going to have capillaries, and we're going to have veins. So the arteries are going to carry blood away from the heart. This is our oxygenated blood. They're going to have thicker, more muscular walls and higher pressure. The arterioles are going to be the smaller arteries, which will branch into the capillaries. So each of these will have very big vessels that get pass down and branch out to smaller vessels and then smaller and then get big again. So capillary walls are only going to be about 1 cell thick. They're very small. The reason why it is helpful for them to be 1 cell thick is because it enables them to exchange gases and nutrients between the blood and the tissue cells. The capillary bed is going to be this interwoven group of capillaries all kind of in one place. And there's really good pictures of this in your book, and I have a few here on the next slide as well. Veins carry the blood back to the heart. These are going to have thinner walls and lower pressure. It's lower pressure because they're further away from the heart, so they're further away from that source. They have valves to prevent backflow. We looked at this in lab. And then venules are going to be those smaller veins, which are then going to branch into capillaries. So here is a little bit of what this looks like. So you have your veins, your venules, and then into your capillaries and your arteries, your arterioles, and into the capillary bed. We also looked at these diagrams in lab. So your blood vessels do have layers of tissue to them, just like a lot of our other organs and body tissues. They have lots and lots of layers. So the first is going to be the tunica intima. This is going to be like our endothelium. The purpose of this is going to be to reduce the friction of the blood flowing through these vessels. The middle layer is going to be the tunica media. And this is primarily a layer of muscle and elastic tissue to help these vessels to be able to change and vasodilate or vasoconstrict depending on what we're needing. And then the tunica externa is going to be the protective outermost tissue layer that just helps to keep these vessels from getting too damaged. So arterial blood flow, let's talk about that. Arteries are going to be under higher pressure because they're carrying blood away from the heart. So because of this, they're going to have way thicker muscular walls to help withstand that pressure. There's going to be elastic fibers in the arterial walls, so they can also stretch and expand to push that blood forward. And here's our last slide. Let's talk about venous blood flow for just a quick minute. Veins are going to be under lower pressure since they're going to return the blood to the heart. So we have these little valves here, and the blood will push up through them, and then they fall back and close. And this keeps the blood from backflowing. It keeps it moving up and up and up, even though it doesn't really have a whole lot of pressure behind it. Skeletal muscles can actually help with this process because skeletal muscles, because they are contracting, can help increase that venous return by putting a little pressure on those veins. That's all that I have for you for your last lecture in cardio. Remember, you'll have 3 lectures this week. You'll have cardiovascular lecture 1, cardiovascular lecture 2, and your blood lecture. So that is all there for you on Blackboard. If you have any questions or anything at all, please reach out to me.